up guys, BIOBEAN here. So for the last little bit, we've been going over some DNA mutations and DNA damage. So today I'm going to introduce two new types of mutations, and then we're going to go over kind of a summary of all the mutations that can occur. So this is a topic that I've already discussed in a previous video, that ionizing radiation can induce double-stranded breaks within the DNA. So ionizing radiation comes from the high energy portion of the light spectrum, going from the very end of the visible light spectrum all the way to gamma rays. And so this ionizing radiation is in the environment all around us, in X-rays, through the sun, and a variety of different chemicals that we're exposed to on a day-to-day basis. So this ionizing radiation can induce double-stranded breaks in the DNA. That DNA is then frequently repaired through non-homologous end joining. And so that non-homologous end joining takes the two broken pieces of DNA and slams them together. And through doing that process, it can actually induce a translocation. So translocation is when a portion of a chromosome gets translocated into another portion of either that same chromosome or a different chromosome. So that could occur with non-homologous end joining, where let's say we have here our broken chromosome in two, and then we have another chromosome just kind of floating around. Because if we think of the nucleus, it's just this really ball of like chromosomes just floating around everywhere in pieces of DNA. So here we have our different chromosome floating around. So perhaps it may be that this is going to get jammed onto the other chromosome instead of jammed back with the original chromosome. So that would cause a translocation where now this chunk is going to get attached to the black chunk instead of into its original blue chunk. So you can get an actual translocation where a portion of one chromosome gets moved to another portion of the chromosome or onto a different chromosome through this non-homologous end joining. All right, so now we're going to get to the last type of mutation that can occur, and then we're going to go into a summary. So last type of mutation that we're going to cover is a mutation that occurs during Meiosis 1. So that is when we're trying to create our gametes in order to do sexual reproduction later on. So during that process, there is the process of recombination. So during recombination or crossing over, the two homologous chromosomes line up and essentially switch information. So here we have our two pairs of homologous chromosomes, one coming from your mother and one coming from your father. So during the process of recombination, they are going to actually kind of switch portions of the chromosome in order to exchange information. So the homologous chromosomes have undergone recombination or crossing over, and they have quite literally switched a portion of their chromosome onto each other. But sometimes this process does not work exactly correctly. Sometimes the chromosomes aren't exactly properly aligned, causing more DNA to move to one of the chromosomes and less DNA to exist on the other set of chromosomes. So let's see what that looks like. So here we can see an example of the chromosomes not properly aligned as they start to undergo recombination. So now when they do their crossing over, this chromosome is going to get a huge chunk of the red chromosome, and the red chromosome is only going to get a small portion of the blue chromosome. So now, because the chromosomes weren't properly aligned, when the crossing over happens, there's an unequal crossing over. So here, this blue chromosome received the A, B, and C genes from the red chromosome, but the red chromosome only received the C gene from the blue chromosome. So now this blue chromosome has 2 copies of the A gene and 2 copies of the B gene, whereas now this red chromosome has no copies of the A gene and no copies of the B gene. So that is what we mean when we say it will lead to copy number variations, because now this chromosome has 2 copies of each gene, whereas now this red chromosome only has 1 copy of one of the genes and is missing 2 genes. It's missing the A gene and it's missing the B gene. So it is going to lead to copy number variations, where this is going to have a higher copy number and this is going to have no copy number of that particular gene. All right, so now that we just went over our last 2 types of mutations, being translocation and copy number variation, I'm going to go into a summary about all the different mutations we learned, how they come about, and what they lead to. So we have ionizing radiation, which is the high energy portion of the light spectrum. And so that can induce double-stranded breaks within the DNA, and that is repaired through non-homologous end joining. So that non-homologous end joining is the slamming together of the two broken ends, and that can result in a variety of mutations. So the non-homologous end joining can result in 4 different mutations. It can result in a deletion mutation, inversion, translocation, or duplication. So a deletion, so if we're looking, here's our original chromosome. It has genes A, B, and C. So a deletion would be a deletion of one of those genes, so just an A and then a C. An inversion would be a inverting of those gene sequence. So now instead of A, B, C, we have C, B, A. A translocation would be moving that piece of chromosome to another spot. So here it got translocated from its original space to now going between after F and G genes. And then we have duplication, so when it's multiplied. So now we have an ABC and then another ABC, so those genes got duplicated. So these are the types of mutations that can arise from ionizing radiation. So the next type of mutation that we spoke about was Tautomers. So that was when a nucleotide will change spontaneously from one form into another. So in this case, it was changing from its normal keto form where it wants to bind with an adenine, and it transformed into its enol form where it now wants to bind with a guanine. So if you're unsure about why that would lead to a mutation, please look at my other video where I go in depth about Tautomers. But now let's talk about what type of mutation that would induce. So that tautomeric shift can induce substitution mutations. So that is in the case where this should have been a TA, but that thiamine had a tautomeric shift, which then induced, after 2 rounds of replication, one of the cell lines to instead express a CG in that location. So it was a substitution of an AT for a CG. All right, next up on the docket is transposons, or transposable elements, or jumping genes. So those are the elements that copy themselves and then move to another portion of the DNA and insert themselves. So that induces insertion mutations. All right, next type of mutagen is polymerase slippage. So this occurs during DNA replication, where either the template strand or the newly synthesized strand slips back as it's being replicated. And that can induce either an insertion or a deletion mutation, depending on whether it is forward or backward slippage. All right, the second to last mutagen is mismatch. So this occurs during DNA replication, where the DNA polymerase is going to put the incorrect base, causing a mismatch. So if that mismatch is not corrected through mismatch repair, that is going to lead to a substitution mutation after 2 rounds of replication. All right, and the last mutation that we're going over can occur during meiosis 1. So my pen ran out, so there's no significance to why this is in blue. I just don't have any more black left. So during meiosis 1, we can get unequal crossing over of the chromosomes, also known as recombination, and that can lead to copy number variation, which we just went over at the beginning of this video. So I've kind of thrown all of those summary slides together into one kind of summary graphic here. So you might be confused that you don't see single nucleotide polymorphisms anywhere on this list. So that is because we call single nucleotide polymorphisms. So you might be confused why you don't see single nucleotide polymorphisms on this list. That is because we call any of these point mutations a single nucleotide polymorphism when we see them in the DNA of people. So as they're actively occurring, we call them mutations, but then when we just look at the total population genomes, we see that there are differences, and so we don't call those mutations, we call them single nucleotide polymorphisms. So even though they are resulted from mutations, we give them a slightly different name, single nucleotide polymorphism. So that could be an insertion mutation, or a substitution mutation, or a deletion mutation, but they all, once we actually see them in the population level or on the genome of an individual person, we then call them single nucleotide polymorphisms. So that concludes the summary video about mutations. And next video, we're going to be talking about meiosis and all the processes that occur within that. If you have trouble understanding this or any other content related to biology, please visit biologybean.com where you can access all of your tutoring needs. And please don't forget to like, comment, and subscribe.